Thank you so much, Mariella, for kind words and for an invitation. Also, thanks, Leon, for hosting this. I really like that core facility type approach. I think it's so important to really sort of bring good biostats, bioinformatics, and also AI into various labs. I'm going to talk for something like 45 minutes, and I'm going to share my slides in a second. But if anyone has a question, I'd be really happy to sort of answer in between. It's like at least when I listen to something for 45 minutes, I just always drift off. So, like, really, just interrupt me if there's any questions. I'd be very happy to not show all the slides, but instead have some discussion. Um,
Sounds good. I will try to moderate. So maybe if anyone with questions either raises their hand or puts their name into the chat, then we can call them out.
well, that'd be great. Thanks so much, Mariella.
Awesome.
And I was asked to talk about sort of recent dynamic type of approaches we're doing. So I was thinking I sort of put it a bit in context. I talk about sort of Velo, CellRank, and these type of things, but then also recent extensions where we go towards more sort of GRN type of learning and a little bit about the sort of more recent AI things at the very end. Very much looking forward to this discussion. Just to have one slide on the place. I think it's always nice to know where people are from. I mean, Mariella mostly said this, but I'm from this place called Helmholtz, Munich. Helmholtz is the largest research organization in Europe. And in Munich, we've been building up a center that sort of looks at various data sets. We call it the Computational Health Center that essentially focus really on AI development and application for faster discovery and translation. And I'm very proud to say that we have more than 40 PIs, more than 500 scientists there. So we're the largest place for this sort of bioinformatics, comp bio health AI type of research in Europe. A lot of dynamics happening. They're really fantastic people, sort of across all the scales. Just last week, one of the sort of senior PIs had a paper in Nature about sort of a model of like a foundation model of cognition that even got into New York Times. We sort of have a lot of grants money. This is sort of just numbers. Maybe it doesn't matter so much. Have a good grad school for PhD students. Try, like, if there's some interest, come to us. We have a European network for AI, strong clinics, a lot of pharmaceutical and tech type of partners. So that's my intro thing. I just sort of need to talk about it because I'm the head of the department, and we're always looking forward to interactions, particularly with colleagues and friends from Boston, where we so often go to, and yeah, always so much enjoy coming. So yeah, let's keep that transatlantic partnership alive. And we actually have a joint thing of sending back and forth people via a joint program with abroad, if you're interested in that. So I was asked to talk about dynamics. So I've been looking into this whole single cell dynamics actually coming from dynamical observation of data. So this is a collaboration with Tim Schroeder, who's now at ETH, who has been sort of looking into hematopoiesis for the longest time, sort of observing cells with microscopy, time-lapse microscopy. Okay, so super tedious to sort of chain these things together and then sort of build these trajectories, these lineage trees of cells differentiating. But of course, the readouts are very sparse. I mean, this is sort of live microscopy. So you have a few markers. There was sort of some endogenous protein, in this case for a decision called the CMP lineage decision. And you saw some markers, you saw them turn on, and you could sort of learn a bit from it. And classical systems biology would be sort of writing up these gene regulatory network diagrams. And we saw these two transcription factors that we actually observed mutually repressing each other. You could see the switches driving it, but turns out this was just maintaining it. So that sort of the dynamics wasn't really predictive, and the type of models that actually worked were deep learning models based on the images of these things. And this is sort of what we put in here. So that was fun. But at some point, of course, we realized we want to go unbiased, and then all the revolution of single cell genomics started, and we've been putting our effort there. But I think sort of piecing together this dynamic information from the static snapshots that you get from transcriptome data, I think is a fascinating question. It's obviously mathematically really cool, right? Because I mean, cells, we kill them when we take them out. Like, how can you put together? There's a few ideas with LiveSeq where you don't need to kill them, but I think this is a dynamic one. Like, how can you put this stuff together? So I think this is the research questions. We have a lot of point observations of sort of cells going somewhere. Like, how can we find information? Like, how can we learn about the molecular underpinning maybe of the observed dynamics once we reconstruct the dynamics and so on? So I try to formulate a few more general questions about this, and this is what I try to discuss in my talk. First, I just want to briefly review, and many of you might know, please bear with me. If not, sort of please ask how to learn these dynamics from snapshot data, okay? Just observe snapshots. How can we then add additional information and modalities? I'm talking a bit about that. How do we do multiple time points? And how can we do this across multiomics and sort of multiple steps in a sort of robust fashion? And last but not least, I talk about how to add genetic perturbations via priors from a GRN, right? So this sort of goes a bit through time, what we developed. And I'm just going to start with the basic part, right? So I'm sure you've all seen Waddington's landscape at some point or another. And I think it's a very nice metaphor, so to speak, of the cell landscape that we can now quite densely sample with single-cell NSA, or to some extent densely, right? And your cells sort of rolling down these hills, making decision at each switching point. I think it's just fascinating. And if we embed data, we maybe see some of these things because we have a lot of information there. It used to be very sparse, but if you now have these sort of very dense ones, you can infer something. And if you maybe choose a correct embedding and a good embedding, in my lab, for example, we've been putting out diffusion maps, which is quite popular for these dynamics. You sort of see them in a range there. And even though you don't have dynamics, you can learn about the dynamics from this observation. This is sort of the core idea of first determining the root and then sort of learning cells dynamics by similarity. This is called pseudotemporal ordering. A whole bunch of papers there. Cole did one of the first ones. Dana, Wanderlust, we did diffusion pseudotemporal, and many others ever since. Let me show you that this is reality. This is not just toy. So in this case, 10,000 or 20,000 transcript dimensions for your viewing pleasure reduced to 2 or 3 dimensions. Now we're flying through the data. I hope you can see it. Each of these dots is now a transcript, like a cell, right? And colors sort of indicate cell types. And you see they're not just randomly filling up the space, but they are connected as was indicated in that sketch, right? And you see, for example, this is a murine intestine. Here's intestinal stem cells, and they sort of make a whole bunch of other stuff. I mean, you can't see that they make it, but you see that, for example, these enteroendocrine downstream cells are connected via these progenitor cells in between. You saw this when flying through, right? And the core idea of this sort of these diffusion-based approaches that we introduced to the field are actually now sort of making a lot of random walks through that sort of landscape in the high dimension, not just in the visualization, and thereby learning then these type of flows. That was some of these very early ideas. A lot of fun math to be done there, sort of learning branching, seeing how this goes, how this scales, and so on. I kind of skip that, but I just want to make the point that, this is useful if you're interested in cells, but I mean, this is a children's hospital, so I'm not sure if everyone is doing omics, but let's say if some of you are interested in imaging, we could actually do some similar stuff. So in this example, it's like an older paper. Each of the dots is actually an eye of a human, healthy or some disease, in this case called diabetic retinopathy. There's various stages. We just arranged the embeddings of retinal fundus image according to this, and we also saw these transitions. You see that? So we sort of see very similar type of human. We could sort of do similar analysis. In fact, we actually recently released a package for doing EHR type of analysis, and not ScanP, but EHR-RP. And we even got into cover of Nature Medicine. That was pretty cool with the method thing. And you sort of see we learn these patient trajectories. If you want to discuss about that, and I mean, Mariella, we discussed briefly, we can actually also start linking this to omics data. I think very exciting. I'm going to take a step away of that and sort of be now a bit more about the method. And the last sort of intro thing I want to show before I sort of tell you a bit more about these trajectory ones is that I think what we've been learning early on is that really the engineering side of things matters a lot. Like to really do this analysis at scale, you really need to sort of sit down. We early on developed this ScanPy, a type of toolbox. At some point, we have more than 5 million downloads or something. It became too much to handle. So we've been putting up this SEverse consortium with a whole set of friends and colleagues. And with this now, I really have sort of for Python an ecosystem that has sort of shared data structures, shared core package, and an ecosystem around this. And we're doing conferences. Next one is actually going to be in US, I think West Coast this fall. We're doing a lot of spatial versions, sort of extensions of that. SquidPy, very popular. Data formats that are based on open microscopy environments, so standards, together with Ollie's lab in particular. And we've been following this up also with perturbation analysis toolbox. Please check this one out. It's provisionally accepted, actually, now coming out in Nature Methods hopefully later this year. And I think it's like, because we have so many data sets now, we can at scale test perturbations, not just like perturbing genetic ones, but other ones. I think a lot of excitement here. Of course, we want to educate about this. So if you want to sort of learn a bit more about this, we've been writing a bunch of review papers, but in particular, sort of having this online living book, which we kind of consider to be best practice and something like that. We're currently extending this towards spatial side in more detail. If someone wants to contribute, this is an open thing, but if you just want to read, let me know if you find this interesting. Okay, taking a step back, let's talk about the dynamics part, right? So you saw so far, we just looked at snapshots of data and connected them. But of course, in practice, I mean, this gives you only so much. Is there maybe more information in the data? And I would say, if I show you now this picture of me from my last vacation in France, wasn't exactly me, but what you can see is I just show you the snapshot of this diver, but you can of course estimate that there's some sort of dynamical acceleration happening. And what I actually would like to know, in physics, if I want to build a model, I don't want to just get state, but you remember that from class early on, you want to get like the speed, the velocity, or the momentum, right? So if I had these arrows, I would see sort of that there's some acceleration happening. And this type of idea, I think, was kind of really cool. There's been a sort of a bunch of approaches to estimate this from time series. I'm going to talk about this in a minute. But in particular, you can also label newly made RNA with nascent labeling, a whole bunch of techniques there. We actually contribute a little bit to this one. Or you can look into unspliced reads. And this is called RNA velocity. It was, I would say, a seminal paper something like 5 years ago from Stan Linnarson and Peter Kachenko. And I think just such a useful, nice idea. This is the idea here. And I want to show you how can we now use this to learn more about dynamics. You know, I was asked to talk about CellRank. CellRank sort of sits on top of that. So just very briefly, the core assumption in RNA velocity literally is that, you know, you have a bunch of data sets of genes, but you don't couple them so far. So it's just really just simple transport model, just very basic central dogma, so to speak. No gene regulation, global shared constant, then you can write up a very simple model. It looks complicated, but it's actually not. It's like as basic as you can get. You can solve it in closed form, right? And, you know, in the early models, you just sort of estimated the extreme ones. We've been extending this with a model called SCVDO that's quite popular, where we sort of now estimate the dynamics there as well. And we could then get rates out, but in particular for each cell, you know, if the cell was up there, if you had sort of more spliced read than unspliced, if you had more unspliced one than spliced ones for a gene, it would tell you that this gene is being upregulated, the other way down. So, you know, you stack all those velocities, and you're like in this phase space, you can now draw an arrow where it would go next. And that's, of course, kind of super cool and useful, right? So that's the idea of RNA velocity. We actually have a deep learning-based extension together with Nir Yosef's lab that we published last year that sort of gives you uncertainty. In addition, technically, I think a super useful thing, and we've been building a lot of stuff on top of that. Sort of, you know, in this paper, we still had a lot of effort to estimate this large-scale model. And here we just throw in a big deep learning thing, and, you know, PyTorch does its way. I think a big change in how we do bioinformatics and this modeling nowadays. In any case, it works well, but I just want to show you some results. So in this case, for example, we look in a collaboration with Heiko Licker's lab from Helmholtz into a developmental process called pancreatic endocrinogenesis. And in this case, you know, you sort of see here these stem cells that are sort of low in this downstream activation factor. They're cycling here, or like this actually. And at some point they start differentiating, this NGN goes up, and they start forming these different cells in the pancreas, alpha, beta, and epsilon cells, and so on. And you see this branching, and you can actually, you see there's some branch overlapping with the others. You can sort of make more sense out of it. But the question, of course, is, so that's kind of cool, right? But can we learn more about it? You know, could we maybe see what the fate of these intermediate cells are? I mean, we can draw these lines, but, you know, I want to see, like, are all of them going up there? Is maybe some going left and right? So what we actually have, and that's sort of the idea of CellRank. I mean, we visualize, also with RNA velocity, we always visualize not just a point cloud, but a vector cloud, right? Sort of a vector field. But that doesn't live in 2 dimensions, it lives in 10,000 dimensions, right? So in there, you know, these things are much more complicated. Can we maybe do more to it? And this is sort of what we do. Like, can we see where these come from? Can we maybe see if what type of genes change across this? This is we can do if we analyze that vector field. And this is sort of the key idea. So we want to go from vector fields that we observe here to really mapping of the fates of cell, of the probabilities, what these different things are. So this is the idea. This is work by, and I need to close my Zoom thing here. This is work by Marius, really talented PhD who worked particularly with Dana Pear's lab from New York, Memorial Sloan Kettering, good friend, fantastic lab, and now is doing a postdoc at the ETH in Zurich. Barbara Teuter's lab. So you have these things, can you sort of learn information there, right? So what we want to do is we want to look at those. We want to identify initial and terminal states. You want to sort of see where do those vectors come from? Where do they go? And we want to assign for each cell in between, you know, what's the fate of this cell to sort of turn left or right? What's the fate of this one? Here it should be very much this, I don't see colors, so I'd say right. Whereas, you know, this one here sort of has sort of both fates, okay? I think a very useful thing. And maybe then you can also learn the underpinning thing. So that's the idea. How we do it is actually not super complicated. Check it out. So this was published here, but it's also a nice website. And it's sort of really nice downstream thing to try on top of this. So what we do is we have this RNA velocity phenotypic manifold, right? So what we do, we zoom in a point. We look at, for example, for each cell, we look at the transition probabilities among the other cells. So they could maybe go, because we have the vector pointing in this direction, and this would be all the nearby ones, we would say, okay, hey, maybe this cell is much more likely to because remember, we have a vector field, right? So this cell is much more likely in that KNN graph to transition to J, and then maybe to some of these. So this we can write up. So we just write up these delta IJs. So just write up a formulation for that. details don't super matter. Now you see, okay, hey, we go more on this. So what we can build these transition type of maps, right? So this is what we do. And so a bit more technically, right? Once we set up this transition matrix, right, we do some type of spectral clustering and some type of cluster analysis. It's sort of a bit like a CellRank type of approach. So once you have this type of matrix here, you want to see sort of what's the probability where most of the cells go if you sort of keep executing this transition matrix. You start somewhere, you do this transition again and again and again, and what's the probability? Where does it end up mostly? It's a little bit like PageRank, where sort of most of the things go with like a bit of an eigenvalue decomposition type of approach. Once we do this type of coarse graining, we see, okay, hey, most of the cells come from here, and they sort of have a metastable state here, and then they go sort of, no, actually come here, sorry, intermediate here, and then they go end up here. So this is the idea. And once you have that, you can then assign what the probability, because you have this transition matrix executed, essentially taking powers of it, you can sort of see where it would go. You get these probabilities here as well. So here it's very sure it goes here versus here, but here it doesn't know yet. So this is what we applied to the questions that I asked on this pancreatic endocrinogenesis situations. For example, are those epsilon cells terminal, right? And what we can do is we can now run this CellRank. We can see, hey, they're coming from here, and they end up in those 3 terminal states. And yeah, indeed, woohoo, as we would know, but we might not sort of see so closely, those epsilon ones are actually sort of sinks of that vector field, sinks where sort of everything flows and then it's going to stop. So these are the terminal states. What fate is there? Well, here you see the fate probability. So for example, all of these ones sort of probable to go here. So instead of what's maybe not so clear here, maybe indicated by color, right? But here, so all of these flowing up here, whereas these ones sort of flow in these other two fates. And where do the beta cells come from? So we could sort of ask, where is alpha coming from? Well, alpha is coming from here, right? Beta is coming from here. So we can really execute these probabilities now. And we can also do a graph representation projecting on graphs where things will go. So that's what you get from CellRank. Are there any questions so far? So I don't see any. So I would just continue, Maria. Is that okay?
Yep, continue. I saved my questions for the end.
Appreciate it. Oh, yeah, don't. I just wanted, like, if people want to interact or something, I'd be, but don't worry if not.
Awesome.
So the question is like, how can we do now? How can we add additional information? In this case, I want to sort of add, maybe time or other type of information. What we now see, so RNA velocity is nice, a lot of applications in literature. But people have noticed early on that it doesn't always work. In many cases, it does not. For example, here we sort of have a limitation paper published a few years ago where we, and this was known sort of by others. This is a data set from Sethi et al. This was actually from Dana's lab. If you look into hematopoiesis, actually these velocities look quite bad. So, this is sort of how normal hematopoiesis should go from HSC, so hematopoietic stem cells, to sort of monocytes and so on. But if you run RNA velocity, the vector field looks like that. So completely bad. You don't even need to start CellRank to see that this is bad, right? So what's the reason? Well, the reason is that, not just the direction is wrong, but, there's all kind of things. I mean, there's enough spliced reads, so it's not that RNA velocity wouldn't work. It should see spliced reads, unspliced ones. But we also see that, these sort of things, these sort of dynamic recursion one of it are kind of misplaced. And if we now exclude, for example, this set here, which is the lymphoid progenitors, it sort of gets a bit better, but still sort of has the directionality wrong. So there's all kind of issues here, and also some of the dynamics are still not very well fit. And reason being that there's sort of a speed up in transcription dynamics here. So yeah, RNA velocity didn't work on this setting. So what do we do? So well-known problem. So we asked now, can we sort of give the method some other type of kinetic anchors informations? Let's say temporal resolution. We're going to look into that later, but maybe give sort of more. And this is the idea of what we now try to do in CellRank 2. So this is work by, in particular, Philip together with Michael and Marius, where we now started to extend this again with Dana's lab to not just go beyond the first kernel set we had in the first CellRank version where we had RNA velocity and similarity, but we now and we used this clustering to sort of give us these type of applications. But now we said, hey, what can we do beyond it? And for this, we implemented a series of other kernels that essentially give us transition matrices. Because once you have the transition matrix, you can just pretty much do the same. And that turned out to be a really useful concept because there's all kinds of ways how you can write up ways how cells transition. So you've been using this type of concept where we just replace, maybe find a new one here, all kind of downstream things ever since. And also just for genetic perturbation modeling and so on, very useful. So what we did was, for example, a pseudotime. Let's kind of obvious, right? But maybe also then an index-based one where, you have a few marker genes that sort of go down like a maturity index. Time series is going to talk about that, metabolic labeling, and so on. And we also implemented mergers of these kernels, because if you have some of these, you can sort of put them together and say, take the joint vector field, weight it or whatever, right? And yeah, we also have some type of test for expression dynamics. So this is the idea. We also sort of updated the sort of whole setup of the package, the number of cells getting more and more, even 2 years ago when we published this. And this is sort of what CellRank can do now with a tenfold speed increase. So this is the idea. And let me just show you how we get hematopoiesis to work now. So I told you, right, for velocity kernel it didn't work. What we now do is we take a KNN graph, just a similarity kernel, but then we add a pseudotemporal ordering one. So we take the distance similarity that we showed you before, we aggregate the two kernels, and essentially just get these adjusted similarities. And with that, we can now get very nice sort of temporal dynamics in a very nice vector field. And we really sort of see now the right dynamics happening, and we can sort of have the right direction in everyone. And we also identify the correct terminal states as you would expect. So this was a really nice workaround, like a very hands-on one. I would say maybe even bioinformatics pipeline one, right? Let's say if one thing doesn't work, let's try another one. And, we have some understanding of the system, make it work. And this is sort of what was happening here. So just to summarize that contribution of CellRank, CellRank 2 essentially extends CellRank to allow other type of kinetic anchors, and, it recovers real dynamics in situations where RNA velocity by itself wouldn't work. And I'll show you in a minute one for additional time points, but if there are questions, happy to take one now. Otherwise, I move to the time point one. I take that as a no.
Yes.
So I just I keep talking. I hope you guys still there.
Sorry, I think we have one question from the audience. I think one person raised a hand. Okay. Do you want me to unmute them, or I think
Yeah, that'd
one
be
question in
nice.
the
That'd be
Q&A.
lovely.
Okay. All right.
You guys want? I mean, don't be shy.
Right.
I could.
Okay,
Krishna, I
perfect.
think you
Yeah.
can speak now. Yeah.
Thanks, Liang. Hey, I have like a couple of questions. How do you actually know what is the truth, right? I mean, I know there you had a ground truth, but in a lot of
Yeah.
data sets, we don't know what the truth is. And how do we actually compare what is either? And the other one is, how do you incorporate like sparseness in data and uncertainty? Just
Yeah.
thoughts. I don't think there is an answer to it, but I'm just curious.
No. I mean, it's a very natural one. I mean, this is sort of the hen and egg, and like a very big problem in bioinformatics, like all the time. Various answers for that. First one, and I think at some point it was totally invoked, then it went down. Now it's coming up again. Is of course you can take simulations. So like really you sort of make your system just work because you know what you put in. Typically it's going to be too simple, or it's going to be not realistic, and people might not care that much, but it's a good thing to maybe convince yourself, maybe your reviewers, maybe your audience at some point that, if your method doesn't work for like a very basic simulation, it doesn't work in general. And for example, we had some very nice simulations for sort of complex hematopoietic branching, super well-studied system. We can write up a GRN and sort of draw from that. In other cases, of course, I think it's safe to say that you're typically never the first one to study a particular system. So there's always prior knowledge, a lot about it, right? And for example, in this one, hematopoiesis is, I guess, one of the oldest studied differentiation systems. So a lot of stuff known. And you can just ask, what happens if I sort of remove maybe one key transcription factor, is maybe this lineage missing, and so on. So these are all things that you can ask into the data sets, like, which are the known downstream ones. So even if you study something completely new, let's say some reprogramming and so on, there's still some basic things that you could expect. And maybe last thing, you also take orthogonal data. I'm going to show you in a minute how we use ATAC in addition, sort of another modality, or maybe spatial. And you can ask on this one. And last thing, you asked about uncertainty or sort of more complex modeling. I briefly mentioned VeloVI. So I think there's some very strong machine learning approaches now to also model the uncertainty of these estimates. I think it's very important. I originally come from math and physics, and I sort of want to have error bars to everything, And with a technique called variational inference, you get at least local error approximations. Global ones are harder, but I think it's a good question. Okay.
Maybe I'll ask my question now then too. Is that okay?
Okay.
Yeah, I'm very interested in trajectories also as they occur in vivo, like ideally in patients, but of course we can't do lineage tracing in patients quite yet, or maybe never. So
people think a bit with somatic mutations, but it's very hard, right?
It's very hard, and especially in pediatric patients, it's really hard because the time is not long enough yet, right, to accumulate a lot of mitochondrial or other mutations. But I was wondering what your thoughts are on the latest, the closest I can get to human, right, is samples from the OR, in our case, brain tumors. and we do spatial transcriptomics now on them, looking into spliced versus unspliced variants, so maybe a little bit infer trajectories. But what's the latest in that field in
Very nice.
finding trajectories?
Yeah, I mean, I mentioned other modalities, and I think it's a perfect fit, right?
On
So
spatial too?
yeah, so on spatial, you can just do what we showed here and do as if just ignoring space, but that's kind of a bit poor, right? Like, how can you take more? I show in a minute how we do with our Moscow toolbox, you use spatial temporal maps. So we build, for example, a spatial temporal atlas of a developing embryo,
right?
Basically merging all of that. But I think the latest thing, and a few people tried this, I think we kind of got it to work now, not for, because I guess what you do might be Visium based in order to get unspliced reads, right?
We do Xenium, so deep data, but not whole genome.
I prefer that quite a bit for some of the modeling approaches, particularly because you get a bunch of transcripts. And what you can do there, you can often localize whether a transcript is
nuclear versus cytoplasmic. Yeah, exactly. I was thinking about that too.
Yeah, and it's kind of an obvious thing. There have been a few papers. We tried this like a few years ago, but data was never good enough. But now I think segmentation is getting there, and actually have one project that we kind of, I think, about to wrap that looks quite strong in that. So if you want to try that out, I think our toolbox seems to be robust enough to estimate nuclear export. And with this, of course, like a directional vector.
Yeah, that's cool.
Together spatial transitions. Really happy to, like, if we can connect about that later, would be lovely.
Oh, that'd be great. Thank you. Awesome.
Thanks so much. And last thing, you can, of course, also use time point information. This is what I show you now, right? So it has been a classical thing, whenever sort of these first type of approaches became more popular, now you can start asking, hey, how can we sort of take more information into account? And, it used to be we have just one, we just do one RSNA. If you had another batch, we sort of just batch correct or just project one. But now suddenly you have multiple time points. And also there, people, we just sort of project into a big map and see where the time points go. But can we somehow prime this model for these time points? There's been a bunch of techniques in literature. I'm going to skip what we did. We did, for example, just sort of, there's a thing called Waddington T. I mentioned this in a minute. We did some other things. Let me show you this popular OT-based approaches. Actually, something that also was developed in Boston was from Eric Landers and Aviv's lab, was sort of in parallel with this, a technique we called pseudodynamics. OT is a beautiful mathematical technique that has gotten quite popular, I think, in our field in particular. What it does is it's now really sort of purely statistical. You have a distribution let's say you have two different distributions and you want to see what's the most efficient way to map this distribution to this one without any prior knowledge. You can add prior knowledge later, but for now, like, what's sort of the minimal change that you need to make, okay? So essentially you say, hey, this cell is going to be transported over there. This is the expression profile. Can I now write down, can I find the optimum mapping of this one to any of the others so that the target one, where it's going to be mapped, is minimal with respect to sort of these weight shifts here, okay? So this is what you try to minimize. And you can sort of adjust the cost here. So this is what you want to do. There's sort of all kinds of regularizations and so on. But this is sort of a core idea. And there's been sort of super many extensions and variations of that. In general, though, a hard problem to optimize, not cheap. But if you can do this, you sort of know how to map across. I think there's one equivalent. So in mathematics in general, it's called Wasserstein distance. But there's some sort of basic, more simpler things where you want to sort of move things with maybe simpler type of distributions. Happy to talk about that. But let me show you what happens with this Waddington OT kernel, and I just want to call it WOT. So let's say we start here with some distribution at the first time point. And Maya, in your case, this could be sort of maybe like a developmental time point or something like that you get in the mice. I don't know if you get it from kids as well. But now you have them. And you, of course, you lose the cell identity. So you never have cell matched ones, but you have distribution matched ones. So the question is, how can you most efficiently, let's say maybe those left cells, maybe higher probability go here than here and vice versa. So how can we sort of map these across? This is how you learn this transport map. And then you can now put these two together, right? And what we could now do is we can now sort of do the same thing now for example, in this case, it was a data set from Shiving. It was a reprogramming data set. It was starting here, was sort of fibroblast reprogramming in different states. And we just ran Waddington OT. We just ran CellRank, but with a kernel that was built from Waddington OT. Okay, so the errors are now not coming from similarity or coming from dynamics, but they come from that sort of state point, time point thing. And then, so those errors point here, we just run it and we see, hey, we see these various circles here. And we can also see sort of what type of influence we have of various factors and so on. And you can see how it links to experimental time and so on. So I think a really powerful approach to put Waddington OT also into the work here. you know, I mean, walking OT has been published before, so I just say we sort of can put those kernels in. We can sort of put time information in addition in. And if you want to keep sort of your velocity in addition, you can just do time and velocity by just taking those two kernels and squash them together, you know, via this kernel fusion approach from CellRank. And maybe just to show you that this actually makes sense in novel data, this was a collaboration with René Meyer's lab from also close to Boston area, a bit west of Boston, where, you know, he has been looking into development of the pharyngeal endoderm, where you sort of see various time points sort of developing here. There's sort of various downstream cell type states that should be produced, and we can run CellRank now on these multiple time points, you know, these 4 developable time points he has here. We sort of identify correct cell states as would be known from literature in these sort of downstream endodermal ones. And we can now also see now what type of genes drive these vectors, right? Because we can see, you know, which gene, if you sort of vary it around, mostly contributes to this sort of shift of a downstream cell type. And we see, for example, there's a bunch of drivers here that are known to be associated to the thyroid lineage and so on. So I think really useful downstream analysis, right? And yeah, and we can then also see, you know, if we sort of combine this with the real-time kernel, so we add this temporal information, we really get the nice lineage drivers. And if you just run Waddington OT by itself, you know, you don't get the right drivers. So it really goes beyond just the Waddington OT because it allows this kernel fusion additional information in there. So we're really happy to pick up a few of known transcription factors and so on. This was sort of validation of what CellRank could do. So to summarize this, optimal transport connects time points based on minimal overall shifts, and it sort of integrates nicely into our CellRank toolbox. So this is my last point on CellRank 2. Now I just want to talk about 2 extensions that we've been doing. One is where we just sort of put this into a much larger scale flexible toolbox to also add spatial information. This is what we call MOSCOT, or multiomics single cell optimal transport. Okay. So this is work by Dominic, Giovanni, also Marius, and quite a few others. A really big collaboration with Heiko Monizan and Marco Couturi in particular, really legend guy for optimal transport at Google in Paris. Or actually Apple in Paris, used to be Google, and it just came out. So what we do here is we could, for example, look in different time points. This was Waddington OT, but maybe also spatial differences and try to see how do things arrange also spatial temporally. You could add some prior knowledge also into this thing, and then you sort of compare those distributions. You know, this is sort of the OT, the optimal transport type of assignment. Once you do this, you can then, for example, interpolate between time points. You can align tissues. You can also do spatial temporal alignment and so on. And I'll show you a few examples of what this toolbox enables. But before I show that, I just want to sort of make a few points. What is really possible with that was not far from possible before. Most of these, actually all of the early methods scaled very badly with respect to time and memory. And, you know, this thing, we sort of were able to make it really linear. I think that's super useful, both in memory as well as in time. So what you see here is a comparison of Waddington OT as an example, and there's many other type of OT approaches versus MOSCOT and a rank constraint, a MOSCOT version. You know, this is logarithmic scale. So this is really big changes. And you see sort of at some point, you can't just even do Waddington OT even for, I think, 50 or 100,000 cells. I mean, there's some new implementations that sort of get a bit more efficient, but at the time was really bad. And you see, this is by far outperforming and maybe even sort of bounded in a linear fashion for these sort of more efficient approaches. We also have sort of much nicer approach where we can sort of combine various of these problems. For example, reconstruction one is NovoSpark and others. And we can do this across multiple modalities at the same time. For example, RNA and attack, also spatial. So let me show you two examples what you can do with MOSCOT. And again, it's just going to be super short vignettes. Please look into the paper. It came out earlier this year. If you sort of want to know more, I'm happy to discuss, of course, about it. But, you know, we know cells don't work in isolation. There's spatial information. So adding spatial transcriptome, of course, is super important. A lot of people thinking about it. And yeah, so happy to hear, Mariella, that you actually also do this sort of in the development and in the kit setting. I think really exciting. What we can do now here, we can sort of map gene as well as spatial position in one joint space. And here we do this, for example, for different organs. Here I see sort of how the liver projects across different time points, what's sort of shared, what drivers here about the brain cells as well. And what we can show is that if we now do this integration, like building this spatial temporal development atlas, this is actually data from a cell paper for embryo development some time ago. We can see that, you know, our OT-based approaches for spatial temporal are clearly outperforming this just time-based one. So adding space really constrains the model more. And here's, for example, temporal mapping of heart cells, and you sort of really see where it would develop. And then you can, of course, interpolate time points. You can sort of extrapolate where it goes. You can see sort of what's driving these decisions and so on. And you can get sort of a heat map of key drivers for each of these developmental spatial steps. I think super useful. I have another example. This was again collaboration on this with Heiko's lab on pancreatic endocrinogenesis. It's the same example that I showed you before, but in this case we had this reporter mouse to tell us where cells sort of would go. We sorted newly ones or sort of positive and negative ones for this reporter line, but in particular also made chromatin. And then we jointly embed those two modalities across those three developmental time points, and we annotate them with sort of various factors for this developmental stage. And what you see is you can now just run your optimal transport across those three time points with a similarity matrix, and you get the typical transition probability as you saw before. This is just a CellRank type of plot, but now based on this MOS called multiomics optimal transport, okay, across modalities. And that's now kind of cool and useful because this you can now query. You can ask, hey, what's actually driving this type of decision? So this is what we do here. First, we ask where are these various cell types now sort of projecting in the next time point? And we see, for example, epsilon progenitors sort of stay some progenitors, but then also turn more into epsilon and so on. But we can also ask in the ATAC profiles, and we zoomed in here in particular in the question, how similar are delta versus sort of substates of delta cells and epsilon cells? Is there maybe a shared lineage of delta and epsilon cells? And is there maybe some sort of substates? We found this subgrouping here. And we could actually validate some of the subgroupings here. I was sort of super excited about that. Just to summarize, I think MOSCOT is scalable, really, and sort of gives OT in the hands of people that really want to work on millions of cells as we commonly get. I mean, I'm sure, Mariella, your image-based data is on that scale. Xenium is really producing a lot of cellular samples and enables a whole bunch of novel use cases for spatial temporal integration as well as lineage mapping. So if I have time and if you give me time, I would now speak a bit about genetic perturbations, but of course, happy to answer questions if there are some for the OT part.
I think we still have time for a few minutes. We'd love to hear about that part.
So in the last minutes, I just want to show you an extension that we've been doing recently that I think is quite exciting because it allows you now to connect these, I would say, quite statistical lineage learning techniques with an underlying regulatory dynamics. And like, what type of priors can you build in? I think the one we all agree on would be the central dogma for sure, right? So that's, of course, just velocity. So how can we couple this velocity where, we assume just no regulation with maybe something more? So yeah, sure, right? With velocity, we know we can get these Waddington's landscapes. We never understand anything about the underlying regulatory dynamics. So there's been sort of the second class of models that learn regulatory networks from these data, but maybe using ATAC, maybe just transcriptome data. And this is a few. Pando was one. The Cell Oracle versions, the Dynamo from Weissman lab. This was from Sam Morrison's lab and other approaches from. So these give you something not about the dynamics, but about the wiring beyond that. Like, how can you now bring this together? And this is the idea what we try to do here. So what we now want to do is we want to now act on the manifold. We want to connect the dynamics to the regulatory network. We sort of have both worlds in one thing. How do we do this? We just write down the equation of sort of cells changing across time. You start with this one. Where would you go next time point? Maybe under perturbation, right? And this then encodes the GRN, and it has a bunch of parameters. And it's just such a big thing that we typically never want to estimate it. So in RNA velocity, we sort of decompose it. We just ignore a regulation. But can we build in a regulation? So this is a challenge. Now suddenly we have to now estimate a 10,000 dimension dynamical system across, let's say, millions of samples even. That's like really hard. Estimate parameters there. It's like something I never dared to try even a few years ago. But with these powerful new network techniques, this becomes possible now. And this is, we call it Reg Velo. Did you see this nice animation? Bring these things together. And this is Ken's idea, or Wei Shu Wang's idea. Really talented PhD who's been now taking this up, taking essentially our velocity type of input, but also a prior graph on regulation, and really putting this together. So for this one, we take the typical embedding. This is essentially our Velo VI model, like just sort of give you this in a latent space and estimate the latent time per cell. But now for each transcription factor here, we sort of put a regulation model in. So for each gene, which by our prior, which ones it has been regulated. And I think there was this question before, right? We discussed about how we add sparsity. There's all kinds of ways how sparsity is being used in these models of implicitly, but here we add it by saying typically there's just a few transcription factors regulating this. So can we write up this regulatory model? We put it in your network, and then we get like a transcription model. We can use ATAC for that. We don't do that yet. And then we just write out the full dynamics of this model. And then we get like a gene dynamics. And we just get a likelihood and estimate parameters. But of course, the problem, as I said, we can actually do it with variation inference. But the problem is now it's a huge ODE that you need to estimate. but this works. And now I just want to show you a few results of that. So, we also discussed briefly about like what type of priors do we have. So one very nice prior for sort of learning about regulatory dynamics is, of course, cell cycle. For cell cycle, we know a lot of stuff. And, there's a bunch of external markers. There's this Fuji time color thing that sort of just gives you orthogonal information based on protein about cell cycle state. This is what we have here. Now we can ask, hey, how do we interpolate this one? So this is what the reg velo dynamics of these data sets look like. And yeah, as you would expect, looks beautiful. It gets you to the right thing. But can we quantify that? Because we want to compare this against previous methods that don't use regulatory network information. So let's zoom in. We get some cross-boundary score. We get some velocity confidence and so on. Now we just combine these scores. And we see our reg velo versus sort of our earlier SC velo and maybe some simpler techniques. This is the best job. I guess that's why I would show you. But it's also most consistent. And it sort of is really good at estimating the latent time. And we can also look at the regulatory thing, which is like not what these methods do. So we can look at the regulatory graph, how has it been updated? Because we don't only just take a prior, but we update the prior. And then we can combine the sort of estimated GRN here versus the other ones, and it's also outperforming. So kind of because it merges these two information is actually better than each of these individual words. So I was really happy about that. Technically, it's a bit hard to use, but I think we now get it to run. So if some of you want to try it out, please just use the notebook or get back to us. And now we can do a whole bunch of cool stuff because now, we just don't have just the dynamics, but now we have an underlying GN, so we can change that, right? So we can take the network and just remove, let's say, a regulon or something like that. Just remove transcription factor. What happens then? So we can see, hey, maybe this lineage is not being made because we moved that particular key transcription factor. What are the effects and how can we compare now self-hate probabilities and what's driving these relative changes? So we can now in silico do perturbation prediction. And I think that's like super much fun. And in the remaining 4 minutes or so, if I may, Mariella, it's okay?
Yeah, can I quickly interrupt here?
Oh yeah, please.
Can you then overlap or feed CRISPR screens into this model?
Oh yeah, absolutely. At the moment, we don't have so much data at scale with dynamical information because we kind of want to add dynamic stuff with it.
Yeah, like perturb-seq information.
Yeah, most CRISPR screens are kind of boring. Like most of them don't do something. Some is just a slight shift. But if you see if you wait a bit and have a longer dynamic shifts, we can actually add that. I don't know if you guys do that, but happy to.
We have longitudinal CRISPR screens like over several weeks, but not yet.
That would be fantastic.
Really self-fate CRISPR screens with a self-fate readout. That has always been my dream, but we haven't quite done it yet.
I mean, we kind of see the self-fate in the RNA-seq, right? So if you have it, so that'd be fine for us. I mean, of course, in this one, the one that I showed you from Heiko Lickert's lab before, we had this NGN color, which is sort of a fate marker, right? Sort of to have this. But I think if you don't need anything orthogonal, so yeah, that's what you can, and you can sort of inform it more. I kind of skipped that. So we have the Reg Velo paper out. So we have an example there where we do this at scale for neural crest development in zebrafish with a colleague from Oxford, where we could actually really in detail do in vivo perturb-seq and then follow up. So we're actually able to validate that, but we can also put it into the model.
That's so powerful.
It's so nice because you can only do so many CRISPRs and you can't do combinations maybe at scale. Now that you have the model, you can interrogate all of that. You can maybe even design like what's the optimal CRISPR to help you sort of do this and that.
Exactly, and hopefully it's going to help us accelerate knowledge about therapeutic discoveries a little bit faster.
Yeah, that's a harder one.
It's a high bar, I know.
We try to combine it with drug type of perturbation screens, but for us it's typically two worlds. We have some things where we can bring a bit together. But at the moment, because you need to add signaling then, right? It's a bit harder.
That's the next part.
All right. I'd love to hear about that dynamic ones. But let me just show you one example. I just take this one again because we've been studying it so much. So just look at the endocrinogenesis again, the terminal states. And what's nice is now if you run Reg Velo, you sort of identify the terminal states earlier with respect to whatever that exactly means. But what's cool now is I can ask, hey, now I know that these alpha, beta, delta, epsilon cells are being made. What happens if I knock out, for example, neuro D2? And you see that, the perturbation effect is sort of strongest in the epsilon cells, as was known, right? And then you can ask, let's screen for additional targets that, this NeoD2 goes for, and you find a few that's sort of known to sort of really have impact for cell fate regulation. And then you can sort of see, hey, what happens if you now do a double knockout of this? And you see then, hey, maybe you get more alpha cells, but then you kill the beta cells as well. So you can ask all these what we call counterfactual. So you use simulation type of questions, and you can then validate them because these things have been studied in the literature before. Or you maybe have an experiment. And I just show you as a last one, again, I skipped the zebrafish one, but in this case, this is not so nicely plotted. We should actually do it on a differential diffusion map, but this is hematopoiesis again. these terminal states, just standard stuff that you know from hematopoiesis, latent time estimation. But now we can remove those two factors that I showed you at the very beginning, this GATA1 and Pu1. You know, those two that regulate the CMP lineage. And you see, hey, they have this orthogonal pattern, just as you would like to see. So it really gives you this toggle switch type of behavior. So I was super happy when I saw this, and you can look at driver prediction, and you know, this sort of really blows many other approaches out of the water. It's much more robust. And you see this is maybe slide, but you see this in other data even quite a bit clearer. So as my last slide, this was sort of now all add prior information to really have interpretable perturbations. But you know, there's sort of this other type of activity, and this is what people sometimes call virtual cells. And I'm still not sure what the digital twin of a cell would be, which is kind of the idea of a virtual cell. A lot of interest there nowadays. Is it something that's only database, or is something where we sort of add this prior information like regular networks so we can interpret? I think the vote is still out there. But the idea of foundation model, I think most of you would know, right? It's like this model that you just train once on a lot of data and then sort of factors to many downstream. So let's say you take all of this spatial RNA-seq and single-cell RNA-seq in the world, train a big old transformer on top of that. Wouldn't that be beautiful to then sort of give you all these downstream type of tasks just under that? A lot of excitement. I don't think we are there yet. Many papers at the moment show maybe the existing ones are not as strong, but there are some, right? And this is from a review we did recently. But I just show you two things that we do get working. We have one where we concatenate disassociated RNA-seq as well as spatial, in particular xenium type of data in a joint model that allows us to then for disassociated one to impute, for example, neighborhood, even though we haven't seen it, because it sort of learns from associated xenium ones. It's just accepted. And we have one, this is sort of these counterfactuals based on a thing called flow matching, which is sort of the hot shit at the moment in AI to generate these fancy images that you see in GPT and so on. So I with this, my last outlook thing, you know, once we have these models, and I put foundation model in, but you know, this could be just as well as these Rigvelo or these earlier models. I think this lab in the loop idea, I think that's really fascinating. And I think it's such a cool one, right? At the moment, I'm always, when I talk to my experimental partners, they have like all these ideas in their head, like what's the next experiment? How can I invalidate this? If we could somehow help support this hypothesis generation, then, you know, to do the experiment and sort of keep updating this model, I think there's a lot of fascination where this would be going. So with this, let me wrap. I talked about RNA velocity, this SCVelos, our CellRank versions, Moscot for the optimal transport, Rigvelo, and briefly about the foundation models. I think I tried to acknowledge most of my collaboration partners and PhDs who did the work. This is our lab in beautiful Germany/Austria when we go on retreats. Please do come by, interact with us. I think we need to keep a connected world, and thank you so much for your attention.
That's awesome. Thank you so much for this amazing talk, really, as always. And each time even more fascinating, but because there, like you said, are new data levels and more can be integrated. So thanks so much. There's a hand up by Sanyi.
Hi, thank you for the exciting talk. These are fantastic. I have two questions. One is that I think CellRank 2 is built on top of the kernel as starting point. I
Yes.
wonder how should people decide what the best kernel to start with? For example, like for time points data, like would it be better to run CellRank 2 on the real-time kernel instead of RNA velocity or monoclonal pseudo time?
Yeah, appreciate that. It's like you give more tools, like how do we start, right? It's kind of hard for us. We often just really start with a pseudotime or similarity one. We have a flow chart in the CellRank tool that you might have seen, Jiang where we sort of try to advise, let's say, if you have temporal information, build it in. If you have ATAC, you know, maybe you can sort of use that modality. So we try to give some advice there. Quite frankly, if you are happy with the unspliced spliced situation that you sort of, because maybe you sort of looked at it, I think it's always a good thing to add. We have in the paper, we also have a metabolic labeling kernel, which I think is super fascinating. You might have seen Weissman, John's lab, and others showing really a good success with that. I guess, of course, Mariela, like you can't label, right? But in some cases, you know, you can sort of get this nascent RNA, and it's like super nice to add. Also, maybe double labels and so on. Yeah, so that's the only thing I can say. But basically, try to get in as much information as you can. But of course, sometimes you want to keep maybe some other information also for validation.
Yeah, thank you. My second question is that if we want to compare fit probabilities like between wild type and knockout, like different conditions, would it be better to do like RNA velocity separately on those conditions, like from early on, Yeah, just basically which step we should separate the conditions.
I really like that you asked that. I think we've been trying to do a few of these, and this differential trajectory comparison, even independent of whether you have sort of velocity added, is not as trivial as it initially looks. Because you should think like, let's say I have this one and then something goes already, like how do I combine this? We've been putting a differential trajectory comparison based on another tool into CellRank 2. We actually made it faster and sort of scalable. Check it out there. I can't remember the name. We call it like the trajectory or something. So that's something that can work. And there we also show a notebook how we do it. But in principle, depending on the kernel that you use, it's going to be not as trivial. Let's say getting robust unspliced and spliced reads to integrate across a lot of very different conditions was not something I think much labs yet achieved. Like we couldn't really integrate this robustly. So if you have that information, but if it's just sort of pseudo-time and so on, you can maybe even before integrate, or you can sort of learn the joint kernel space first and then do differential comparison, or you sort of learn them separately. Ideally, you have everything in a joint map. So that's always easier to compare. So we try to put everything ideally in joint coordinates, but if it's got unspliced reads, we weren't able to do so. So these we calculate separately.
Yeah, thank you.
Appreciate it.
Thank you. And Krishnan has his hand up. Do you want to unmute yourself?
Yeah. Hey, quick question. So for Rechwell, I was just wondering, let's say you have pre-end states, and if I say, I'm wondering about the inverse problem. Let's say if I just want to, in case of say, I would just want to have enhance, state C instead of say B, So reverse, so can you do like a centrality and can you see where should I actually perturb?
Man, I really like that. And this is when you remember I talked briefly about the experimental design. This is sort of one of these questions, right? Like how can we, now that you have this projected end state, this is sort of losing all that dynamic information before, like how can I enrich the one versus the other? And I was talking to Mariella briefly before. We have another thing where we look into various protocols, how to make organoids, in this case, brain organoids. We made a brain organoid atlas recently. And there we also asked, like, how can we enrich this one hindbrain cell type that apparently seems to be not in those organoids, that's sort of missing in the versus the primary reference? Like, how can you enrich this? These are really interesting in what's in math called inverse problem. I think you sort of used that already, right? And there's various ways how to do this. So what we do at the moment, quite frankly, is because our condition space is often not too large. Let's say you just have to do 1,000 knockouts. This stuff is easy to simulate. You just do each of them and see which one is sort of closest, okay? But let's say you now have also time points when you do that. Let's say maybe you also have concentration. Suddenly this space to screen gets so large that just a combinatorial exhaustive thing doesn't work anymore. And then it gets really fun. Then you get in this range of, maybe call it even not just active, but reinforcement learning, like a search in that perturbation space. And I think this is like a really hot topic. Actually, I have a very mathematical student currently working on solving this inverse problem in this OT space. Like there's some nice math to be done there. We're really shopping for good data sets and questions where people really have such a combinatorial space. It turns out in many cases, a rough discretization still does the job. Like for example, in this organoid one. I mean, I always found it so fascinating. I don't know if you know much about these projects. I just did before, but people sort of pipette in stuff and do medium change at kind of random time points. Like I don't know where this comes from, right? It's just like someone established it, kind of works. Yeah, let's go with that, right? And you sort of vary a bit. So we just discretize in like seven day intervals, and then we search this discretized space. So that's kind of okay. But of course, at some point you really want to fully optimize, and and then of course you need uncertainty for that, because whatever you predict, you're not so sure about. But I think this is going to be the next thing. And this systematic experimental design, of course, not to say, hey, you need to do that, but just give sort of some hint to your next selection of, let's say, candidate drugs, candidate CRISPRs, candidate time points, and so on. I think it's really fascinating.
Do you think we can actually use something like eigenvector centrality or something? Because in some sense it is, to find out what is the central node, and that's all.
Yeah, it's usually not that easy, because let's say if it's a graph or so, sure, right?
Sure.
You could do vector decomposition and so on. But typically the underlying structure that you need to search is typically multimodal, has a bunch of different covariates and so on. So yeah, and these are strongly nonlinear models. So I think such linear approach is not the way to go. Maybe as first approximation, maybe for interpretation, but we didn't do that.
Sure.
Awesome. Maybe one more question from my side, unless anybody else has one. Fabian, I'm really also therapeutically interested in end stages, right, of differentiation. And my main question there is how much of an end state is it? Or can it ever go back? In terms of cancer therapeutics, most is where I'm coming from, right? And so we are all trying in the lab to look at what are the differences in the end stages that stay there and the end stage cells that at some point do go back? And I wonder, and we're not quite there yet, but I wonder what we actually need in terms of data generation now that we could then feed into a model that could help me then predict later on.
Like a revertibility type of score?
Yes, exactly. Because, differentiation therapy or maturation therapy is kind of hot in oncology, but many people doubt that cells will really stay there once you push them that way.
Okay, I see. Yeah, so we've been not so much looking in the oncology space, but we've been looking in this diabetic space. You can nowadays kind of symptomatically treat diabetes, right? But you can't like generate beta cells so easily. So this, and one observation we have together with this colleague is that they also revert back to sort of a de-differentiated state in obesity or something like that, or in also diabetes. So question is like, how can you make them sort of become differentiated again to be functional,
And stay there, yeah, and not go back.
Yeah, and I mean, we haven't. So essentially what you need to do, right? You need to sort of build a model of these two cases, like once there's de-differentiated, once there's sort of a bit more differentiated, and you can sort of see, is that sort of a deep valley in that Waddington thing, or is there sort of a little outlet where they can sort of still swap over with a little bit stochastic fluctuation?
That's right. And I'm wondering what we need to even get to making a model, right? Do we need barcoding and single cell over time as a readout to know like what this little niche might be?
I mean, so is the observation that they just sort of revert back over time, or they revert back because they see some stimulation, some signaling thing or so that sort of makes them like, do you know if it's context or if it's really just like random internal fluctuation that makes
We don't know. That's exactly one of the questions, right? And I think every single cancer type is a little bit different, so it's complex, but
if can you isolate cells and let's just like in vitro see if they revert back, or is that too toy?
You keep them long enough and then a few go back. But
okay, a few. This is so cool, right? These things are so plastic.
Yes, and I don't want them
cool.
to be.
I mean, it's kind of bad in this, right? But it's fascinating.
But how to build a great model so I can maybe predict better and say like, only if you have this external stimuli, or only if
I think with the model we can ask, so because we have fate probabilities, right? So of course, like you have the probability for going back, right? So you can now ask, hey, what type of genetic perturbation do I need to do? Let's say, because at the moment we have been sort of working out with CRISPR, right? Let's call about the molecules a bit later. What type of CRISPR can we do so that this fate probability here goes down, so that it doesn't move? So this is, I think, the question we could do. Yeah,
that's
I think
very cool.
this regular should enable this one. Yeah, but I'd be lovely to explore a bit more. And ideally, maybe we don't take a fully unstudied system, but some, people have been maybe even a toy thing where you can sort of see it fluctuating back and forth and you just make it not go.
That's right, with the color, yeah. Well, a lot to think about, but thank you so very much for this very stimulating talk, and we're all excited about what's to come, from your lab, but from all of us as a community as well as we continue to work together. So thank you for talking to us today. It's really awesome.
Really appreciated the fun discussion. Thank you as well.
It was really great. Thank you.
Thank you both. Yeah, thank you both. Thank you all the people joining us. Good discussion. Have a nice day.
Thanks so much.